Hello, everyone. My name is Tremaine Whitehead, and my presentation is on plant domestication, and this is for Hort 524, Professor Thompson. Hello, everyone. Today I'm going to talk about plant domestication, specifically the plants that we eat every day. As you see on the slide, we have on the left, which is teosinte, which is the ancestor to the modern corn that we eat today, which you see in the image on the right. We often take for granted that corn comes on a cob, wheat grows in neat rows, and rice is harvested by machines. But none of this happened naturally. Every fruit crop we eat today started as a wild plant that humans gradually and sometimes dramatically changed over thousands of years. In this presentation, I'll cover over three main things. First, what made certain plants domesticable in the first place. Second, the universal changes that occur when a plant is domesticated, both the benefits and the trade-offs. And third, where domestication could be headed next, including CRISPR gene editing and perennial grains. Here's a surprising fact that I found out. There are about 7,000 edible plant species here on Earth, but humans have only domesticated around 200 of them. And just 20 crops, things like wheat, rice, corn, soybeans, and potatoes, provide 90% of the world's food. So it makes you wonder, why those? Why not the other 6,800? This is what researchers call domesticability. The idea that some species were simply genetically predisposed to being domesticated. According to work cited by Biernick and colleagues in Frontiers in Plant Science in 2025, plants had traits that made them respond more quickly and predictably to human selection. These include high phenotypic plasticity, ooh, meaning the plant could change its form easily, favorable mutation rates, and what's called simple trait linkage, where a desirable trait wasn't tied to undesirable ones. Now, when a plant does become domesticated, it goes through a set of predictable changes known as the domestication syndrome. These changes show up in almost every domesticated crop. Seeds end up getting larger, grains stop shattering, meaning they stay on the stalk longer instead of falling to the ground, which actually makes harvesting possible. Dormancy may be reduced, so seeds germinate uniformly when planted, and plants develop a more compact growth habit, which allows farmers to grow more plants in less space. Cow and colleagues writing in iScience in 2025 show that these changes are written in the genomes of crops. So they use genomic analysis, which can actually see the specific genes that were selected, uh, genes controlling size, flowering time, and branching patterns. A key review by Hong and colleagues in Cell synthesizes how genomics have transformed our understanding of domestication across dozens of crops. And and as G explained in Molecular Plant 2026, even rice, which we think of as a single crop, was domesticated at least twice. Once in Asia, which is the main rice that majority of people consume, and then once independently in Africa. African rice has its own unique domestication history with different genetic signatures. This tells us that the domestication syndrome is not a single path, but a set of solutions that different plants arrived at independently. So something we can take away from this is that domestication wasn't random. Certain plants were pre-adapted for it, and once selected, they changed in predictable genetically encoded ways. And so now we'll look at what those changes actually do in the plant's biology. Domestication is, at its core, about one thing: getting more food from less land. A meta-analysis of 54 wild and cultivated species published in Crop and Environment in 2024 found that domesticated plants grow 30 to 50% faster than their wild relatives. They have higher rates of photosynthesis, their leaves are larger, and they allocate more nitrogen to the parts that we eat: the seeds, fruits, or the tubers. But these gains can and do come with trade-offs. Secante and colleagues in a 2020 study of 5 wild edible herbs like dandelions and plantains found something fascinating. When these plants are domesticated, they often lose many of their chemical compounds that made them healthy in the wild. Things like phenolics, flavonoids, and vitamin C, all of which are antioxidants and have medicinal properties, drop significantly. The domesticated versions are less bitter and more pleasant to eat, but they're also less nutritious in some ways and have fewer natural defenses against pests and diseases. This brings us to a more subtle point about how domestication works at the molecular level. Ballum and Bendamin, writing in Molecular Plant in 2025, studied cucumber domestication and discovered something unexpected. They found that some domestication traits are controlled by what we call silent mutations. These are changes in the DNA that do not alter the protein sequence, but do change how genes are regulated. These epitranscriptomic changes, modifications to RNA, can have dramatic effects on the fruit size, shape, and even flavor without changing a single amino acid. So domestication is not just about big, obvious gene changes. Sometimes the most important changes are hidden in the regulatory code. And then there are crops like sugarcane. Garsmer and colleagues in a 2025 paper in Cell analyzed the genome of sugarcane, which is an incredibly complex polyploid, meaning it has multiple copies of every chromosome. They traced its domestication back to wild species of saccharum and showed that modern sugarcane is actually a hybrid of 2 or more wild species. This kind of hybridization, where wild relatives crossbreed and humans select the best hybrids, is a recurring theme in domestication, especially in crops with large, complex genomes. So when we look at this, one of the takeaways we get is that domestication delivers real yield gains: 30 to 50% faster growth, higher photosynthesis, more edible biomass. But those gains come with a caveat, and they can be lower natural defenses, reduced antioxidant levels, and sometimes a loss of medicinal compounds. And the molecular mechanisms can be surprisingly complex, from silent regulatory mutations to whole genome hybridization. So we will look now at a specific case study that shows how we can actually see the changes in the genome. We will now look at a real-world example to show how modern genomics allows us to do something that was impossible merely a decade ago. We will separate what humans deliberately selected from what the environment selected on its own. The example comes from a 2025 study on eggplant. Uh, though I'm drawing on the principles demonstrated in the Almandi study, which analyzed over 3,400 eggplant accessions from across Asia and Africa. Using genome-wide association studies, researchers could identify which genetic changes were driven by human choice and which were driven by climate and geography. What did humans select for? The things that you would normally expect. Fruit color, specifically purple and the black eggplants, because to them that's visually appealing. We also did loss of the prickles, which is the pricklies that you find on the stems, because no one wants to harvest a spiny fruit. Larger fruit size, obviously, and reduced bitterness, because bitter eggplants are less marketable. Now, what did the environment select for independently? Flowering time, which is plants in different latitudes need to flower at different times of the year. Drought tolerance, which is crucial for plants grown in monsoon areas versus arid dry regions, and disease resistance. Different pathogens exist in different places. So this is one of the surprising this is a part of the find that was surprising. These two sets of traits, the human selected versus the environment selected, were located on completely different regions of the eggplant genome. There was almost no overlap. This means that humans and nature were pulling the plant in different directions on different genes at the same time. So domestication isn't just humans directing evolution. It's a negotiation between human desires and environmental constraints. And then as a second example, we consider the allium crops, which are onions, garlics, and leeks. A 2026 review in iScience showed that these crops have some of the largest genomes in the plant kingdom, with over 90% of their DNA being transposable elements, which they call jumping genes. This repetitive DNA makes domestication slower and breeding more difficult, but it also creates enormous chemical diversity. The sulfur compounds that make onions pungent and garlic medicinal, compounds like allicin, are products of domestication selection. Humans chose for stronger flavor, and in doing so, they also selected for cardiovascular benefits that modern science is only now learning and understanding. So, taken from this, we see that genomics let us see domestication in exquisite detail. We can separate human choice from environmental adaptation. We can see that these forces act independently on different genes. And we can study crops with different genomes, like onions, and still trace how human preferences shape their chemistry. And so now we looked at the past and how our corn has come from the past to what it looks like now, things that we have looked at in the present. And so let's see now how the future of plant domestication could look like. For most of modern history, domestication was slow. It took thousands of years to turn teosinte into modern maize and wild mustard into broccoli, cauliflower, kale, and cabbage. But we are no longer limited by that time scale. The first example is what we call neo-domestication, which is taking a wild, undomesticated plant and using gene editing to rapidly turn it into a crop. A 2025 review of in theoretical and applied genetics focuses on wild pigmented rice, sometimes called forbidden rice, which is packed with anthocyanins, the antioxidants that give blueberries and purple grapes their color. This wild rice is incredibly nutritious, but it has terrible agronomic traits. It's tall and falls over in the wind. It shatters its seeds onto the ground, and it takes a long time to mature, and it produces very low yields. Using CRISPR-Cas9, researchers are now editing just 4 to 6 key genes in this wild rice. They're inserting the semi-dwarfing gene that was so important in the Green Revolution. They're knocking out the shattering genes, and they're shortening the life cycle. And crucially, they're leaving the pigment and nutrition genes completely untouched. The result will be a new ancient crop, one that combines the nutrition of a wild plant with the yield and harvestability of a modern crop. And they can do this in 3 to 5 years, not 3 to 5 millennia. The second example is even more futuristic, and that's perennial grains. Almost all of our grain crops, which we know are wheat, rice, corn, barley, and oats, are all annuals. You plant them, they grow for one season, you harvest, and then you till the soil and plant again. The annual cycle is a major driver of soil erosion, carbon loss from farmland, and water pollution from fertilizer runoff. Every year we lose billions of tons of topsoil because we keep replanting annual crops. A 2025 review in Frontiers in Plant Science make the case for domesticating perennial grain crops. These are wild relatives of existing grains that live for multiple years. They have root systems that go 10 or more feet into the soil. These deep roots hold soil in place, sequester carbon, and draw up water and nutrients from deep underground, reducing the need for irrigation and fertilizer. The challenge has always been that these perennial relatives are hard to breed and genetically transform. But the paper describes new in planta transformation methods. These are techniques that don't require growing cells in a lab, and they are finally making perennial grain domestication feasible. The goal is to have the first domesticated perennial crop in farmers' fields within 10 to 20 years. Now imagine wheat that you planted once and harvest for the next 5 to 10 years with roots that would build soil instead of destroying it. So a takeaway or the big picture that we get from these type of topics in this whole presentation is this: domestication is not ancient history, and the conclusion, it is an ongoing act of science. We are still domesticating plants, but now we have tools like CRISPR that compress millennia of selection into a few years. And we are setting our sights on entirely new kinds of crops, like the perennial grains that could transfer agriculture from a soil-destroying process into a soil-building one. The plants on your dinner plate are the products of thousands of years of human selection, written into their genomes. And the next generation of food plants, the ones your grandchildren might end up eating, are being designed right now in laboratories and field trials, using the same principles of domestication, but with 21st century tools. That is something. That is really something to think about. And I hope that everyone got something from this. And me, myself, it really makes you think about what is capable out there with plant domestication. Like, it really makes you wonder what the future looks like. Thank you very much.